Hey, this is Joel Duff. Welcome back to the channel. Have scientists been pulling the wool over our eyes for some 20 years? And when they make statements about the similarity between chimpanzees and humans or other great apes and human beings, using numbers like 98 or 99% similar in their sequence, is that is that a fraud? Because here on the cover of Answers in Genesis, the front page of Answers in Genesis website, we have Ken Ham saying that study finds chimp DNA not 99% identical to ours. And according to Ken Ham, this is shocking to other scientists. This is shocking to the world. All right. Because there's a new study that just came out that confirms exactly what Ken Ham believes and expects from the Bible, which is that chimpanzee DNA could not possibly be only 88 or 99% identical to human DNA. And therefore, this study that shows it isn't is evidence of his conception of the relationships between species, or the amount of differences that are required to call two things different species. All right, newsflash, not shocking. This report, this paper, not shocking at all. I'm going to show it to you. We're going to do a couple different things here. We're going to read this article, most of it, so we can get an idea of like how Ken Ham is portraying this news to his audience. We'll take a look at his source, which is Evolution News, which sounds like it would be news from like an evolutionary point of view, but no, it isn't. It's actually an intelligent design, all right, website that reports on news about evolution, but of course it's all about reports about how evolution is wrong. And then we'll take a look at, I don't know what order we'll do this, see what happens. We'll look at the actual Nature article, which is a article that reports on the complete genomes of multiple different great apes, right? And comparisons of those. And so, of course, one of those comparisons is how genetically similar different apes are from each other. Notably, I'll just point out, Ken Ham's going to focus totally on chimpanzees and humans and the similarities between them, but we might want to note how similar a chimpanzee is to a bonobo and maybe a chimpanzee to a gorilla, to get some kind of perspective, right? Some kind of apples to apples comparison, to get an idea of like, what does it mean that there's a 14% difference or a 2% difference or 1% difference or a 20% difference? Does that mean anything? What does that mean? That's really the question here. What does 99% identical mean versus what does 14% different mean when you say genetic similarity? And do we all mean the same thing when we use those terms? All right, that's a good part of the story, is we're not always using the same terms in the same ways. So we'll do all of those things and maybe look at a couple other sites too, to look at genetic identity. So we got to get cracking. All right, so here we go. Study finds chimp DNA is not 99% identical to ours. From Ken Ham's blog. Doesn't mean that Ken Ham wrote it. He has the assistance of a writing team who put this together. Here is the article. Another article we'll probably look at that is his source, right? Bombshell. New research overturns claim that humans and chimps differ by only 1% of DNA. Overturns. Loves to use that word. This is from Evolution News. So a shocking new study has confirmed exactly what we expected, starting with the Bible, what creationists have been saying for decades. Chimpanzee DNA is not 99 or 98% identical to human DNA. Okay, well, that's not actually shocking to us. Who do you think is shocked by this? I have yet to see any shock and awe that's come from this paper. This paper came out. It was like, it's a cool paper. I looked at it. I was like, awesome. I'm really glad that they have that data, that we have the complete genomes, or at least much more complete than they were before. Just like the human genome was completed, some 20 years ago, but it wasn't really quite complete because there's like a lot of little gaps in there, and it's only been recently like completely complete. All right, with actually there's probably still a couple little tiny gaps, but nothing like there was before. All right, and another key theme here is going to be that's, we have like that one complete sequence, and now we've completed other one. So what is the human genome? Well, we're all different from one another. We're going to talk about how different we are, because that's a very important part of this story. If you're going to compare humans and chimps and ask the question how similar they are to each other, when you get a number like 14% or 8% or 20% or whatever you're going to come up with, the question you really want to ask is, how different are you and I? So relative to that difference, how different are you and I? I can tell you what, based on the calculation that evolution knows does, all right, you and I are a lot more different than 1%. Okay. And so even human beings aren't 98 or 99% similar based on the way that they're going to calculate this percentage. Now, the headline in the paper of Nature is rather bland. Yeah, the Nature paper doesn't say like, evolution overturned, or like we didn't know what species were, or like genetic similarity is radically different than we thought. None of those things. And I'm sure those authors never even thought to say that. And it wasn't because they were trying to hide something. All right. They weren't trying to like pull the wool over our eyes one more time. Right. The title is Complete Sequencing of 8 Genomes. And I'll show that to you. Hey, you know what? Let's just do that right now. So boom, there you go. Complete sequences of 8 genomes with a whole lot of authors, because this takes a lot of work. This took a lot of work from a lot of people, and everyone who contributed to that ends up everybody who, either did, help extract DNA or get access to the samples, to all the people, the technicians that helped sequence this. So you can see they did a bunch of different great apes, all the major, representative from all the species of great apes, and the major subgroups of different great apes. Really quickly over here, we get the total number of bases, like around 3 billion base pairs as part of one copy of the genome. And human beings have around 3 billion base pairs as well. But let's wait before we get to that kind of stuff. All right, back here. They should have titled it Evolutionary Ideas Wrong for Decades. Humans and Chimps Vastly Different. That would be a terrible title. It really would be a terrible title. There's nothing about this paper that says that evolutionary ideas are wrong. Ken Ham's conception of what genetic similarity is and how it is used and defined by different evolutionary biologists is wrong. Human and chimps, vastly different. Well, now we're going to ask, like, if humans and chimps are vastly different, what other things are vastly different? And we're going to end up saying things like coyotes and wolves are vastly different because they're about as different as chimpanzees and human beings. Some human beings might be vastly different from each other, compared to what Ken Ham might have thought. In fact, I think Ken would be rather shocked at how different human beings are. And don't get me wrong, human beings are actually amazingly similar to one another. But in terms of the overall size of their genome, you and I could differ by up to 300 million base pairs, right? You might have 300 million base pairs of sequence that I don't have, right? That's some, 9% difference if you count every single one of those base pairs of that 300 million as something that I'm missing. And therefore, every one of those base pairs is a difference between us because you have it and I don't have this base pair. You have that base pair. I don't have that base pair. That's 9% difference. So if you'd like to count differences that way, you and I are multiple percents different from each other in the totality of our genomes, or at least in terms of the extent of the total number of bases in our genomes. In addition to the individual bases that may have differences, like you have an A and I have a T at the same position. Right. So that's where we're going to be going here, is we're going to look at that kind of stuff, like how the accounting of these differences. So are humans and chimps vastly different? I'm not denying that they're not quite different, clearly different species, right? And there are clearly very significant differences between those two species. But how much of that is attributed to, genes and the actual sequence itself? I mean, that's something that's, being studied all the time. We're trying to figure that out. They don't like to draw attention to the false narrative they've led the public. In fact, they don't even clearly acknowledge their evolutionary expected findings in the paper. There's nothing about evolutionary biology that would suggest that humans and chimpanzees have to be like almost no difference in their genome, especially since geneticists have recognized that there are large differences in genomes between all different species. Right. And there's some exceptions, but for the most part, a well-developed species that's different than another species, even if they're closely related, are going to have millions and millions and millions of differences. And based on the accounting that this group wants to do and that what Cunningham wants to do, they would have potentially significant amounts of like repetitive DNA, which are just repeat sequences, for which one individual might have 100,000 repeat sequences, another one might have just, 10,000 repeat sequences of, say, 10 base pairs. You add that up and you're looking at, a million or two base pairs difference between those organisms. But does that really make those organisms that different? I mean, it does just in terms of like the amount of actual raw material there of A, T, C's, and G's. But what are you doing with those A, T's, and G's? That's really what's more important. And that's a much more difficult question when you want to ask the difference between species, what accounts for it, what variants are actually important, and what differences make no difference. Okay. The fact that you have several million base pairs that I don't have, or that I have several million base pairs that you don't have, does that mean that you're not a human being? Does that mean that you and I can't be related to each other at all? That we're significantly different, that we're vastly different from one another? I would say no. I would say like actually the difference between us is extremely small. I'm totally comfortable with that idea, like less than 1%. Because it's less than 1% of the actual variants that make up the real differences between us, which are individual variants in different genes and regulatory sequences that change how how we're formed physically. Whereas you could have an extra million base pairs that does nothing, right? And the fact that you have those and I don't have those makes us no different from one another, other than you're carrying around some extra base pairs that you have to copy that I'm not copying. So we need some apples to apples comparisons, not apples to oranges comparisons. What they did in this study was provide complete sequences for 6 living species, including chimpanzees, bonobos, 2 species of orangutans, gorillas, and a gibbon species. What's unique about this study, according to Evolution News, now once again, they're using their source, according to this source, which is highly biased, written by an author who has a PhD in geology, right? And doesn't have any significant real experience with genetics and evolutionary biology, but writes about it all the time on Evolutionary News, and has a propensity for being quite flamboyant in his ability to misunderstand evolutionary theory. I'll just put it that way, right? That's the source that Cunningham is using rather than referencing the actual paper itself. Okay, so how does Evolution News, which I think to the audience of Young Earth Creationists, if they're reading this article and they don't know anything about Evolution News, which most people wouldn't, right? It's like, oh, Evolution News said this? That sounds like a site that would be supportive of evolution. That's exactly why the site's named that, Evolution News. It's to suck people in and think that this is a site where you're going to learn about evolution, and that that is not an anti-evolutionary site. Right. So Cunningham can readily, reference this particular site and get his audience to, because they're not going to click on it. They're not going to try to understand that site and who's behind it and all that. Right. They're not checking their sources. They just see according to Evolution News. And so, oh, this sounds like a secular source. And look what this source is saying. This team of researchers has published complete sequences of ape genomes that were created from scratch rather than using human genome as template. As a result, for the first time, we can attempt a much more accurate assessment of the true degree of differences between human and chimp genomes. Many of the genomes assume common ancestry, and then use the results to prove common ancestry. It's circular reasoning. I don't want to get into the whole like how they use the human genome as a template, because we simply had the human genome done, and we scaffold or overlay the sequences we get from other species on that to find where the similarities are. And then the thing is, not having a native original sequence, it's hard to find where those, where the stuff is in the gaps, especially repetitive sequence. It's hard to like piece that together. So what they've done is they've started from the ground up, and they've said, we're going to do, because there's a lot of new sequence technology where you can do really, really, really long sequences with technology where you're directly sequencing the bases themselves rather than having to copy them and do a whole bunch of intermediate steps. So as a result, it's really because of technology that we're able to get this kind of data today. It's not necessarily an evolutionary assumption. It's not something somebody was doing as sort of an end round in order to like fake the data. It's simply convenience, right? It was extremely expensive and hard to do. And so you have a human genome. And then the chimp genome is built in a similar fashion, but because there was so many gaps and it's so expensive to try to fix it, we've never really had like the complete chimp genome until we have better technology. So what did they find when they did actual observational science? I mean, these are all just catchwords thrown in to make it sound like previous scientists were not really doing the right kind of science. And now they finally caught up and they're really getting the real data as if the data before were invalid. Rather than the evolutionary storytelling, again, they don't have I would need evidence for how they've manipulated the data through this method rather than just innuendo. Well, they found what creation scientists have always been finding in their research. Chimp and human DNA is not very similar. Now he's referring to like Tompkins' work at Institute for Creation Research, where he's been saying for a long time that humans and chimps are only 70% similar. And then they found an error in this calculations, and now it's like 88% similar, sometimes 85% similar. So big, big differences. Now, he has a really quirky way of adding up the differences. And, I've called him out many, many times. We've had this discussion a lot of times about how he's never really applied his methodology to comparing like chimp and bonobo, or comparing chimp and other primates, or even comparing one human to another human, or the human to the Neanderthal sequence. And if he did that, he might find that, you know what, using that method, humans and Neanderthals might be 10% different. They might only be 90% similar. So the whole like chimps and humans are 85% similar. It doesn't sound nearly as dramatic, does it? Right? And see, they love to talk about the 1% because that sounds like, it used to be 1%. And now we're saying, as we're going to see in a moment, they're going to say like, well, now this new data suggests it's 12 or 13, or even if you add it all up, 14 to 15% different. That's pretty close to the 85% that Jeffrey Tompkins had. So maybe he's been right all along. 15% is so dramatically different than 1%. That's a massive error. There has to be some fraud involved, right? In order to be that different, to be that far off. Scientists clearly had to be hiding this fact. And that is why it is so shocking. Except no one's shocked, right? Because if you know what's going on, there's nothing shocking about this. Nothing. Totally expected. Might be a little bit more. I don't know. I'm not shocked. Let's see what the Evolution News. This is, again, Evolution News summarizing that paper from Nature. So in their estimation, at least 12.5% and possibly up to 13% of the chimp and human genomes represent a gap difference between the two genomes. That's just like what I just said. If you have 300 million bases or 100 million base pairs of actual code, sequence, I don't want to say code, of sequence, A, T, C's, and G's. All right. In your genomes, I got 50,000 base pairs on chromosome number 2, and I got 75,000 base pairs over there on chromosome number 3 that I have that you don't have, right? And maybe I've got 100,000 base pairs of sequence one place that you don't have at all, right? Is missing from your genome, right? Each one of those would be gaps if I compared my sequence to your sequence, right? If I lined your sequence of your chromosome up to mine, it would be like there'd be a missing place. A missing spot in my genome. And sometimes you'd have a giant missing spot. So they don't align. If they don't align and you count every one of those gap spots as a difference, well, then that's how you're going to end up with hundreds of thousands, actually millions of differences between you and I, and potentially 100, 200. And I'll show you that there's an example of 300 million differences, which would be 8 or 9% difference between two human beings, right? And this is already known. Okay, this isn't like, that's not shocking to anybody either, because that's already been reported. Okay, so if two human beings can differ by many percent when you add up, when you calculate this way, which Evolution News is doing, this is how they're calculating it, the difference between chimpanzees and humans, then wow, how shocking is it then that you end up with 15%. There's also significant alignable sections of the two genomes that show short nucleotide variations, which differ by only 1.5%. See, all the sequence that it's aligned, like all the genes that organisms share, right? And all the really, I'll say the important stuff. If you line that up between two different species, like chimps and humans, between dogs and wolves, wolves and coyotes and foxes, lions and tigers, right? You line species like that up with each other, and you line up all the sequence that they both share, and you ask how many differences are there at the individual sites. And what you get is less than 1%, maybe 1.5%. The numbers that we've been throwing out for a long time about the similarities between species. Right, so if you add that to the 13.3, then you get up to 14.9%. Right, this means that the actual difference between human and chimp DNA is 14 times greater than the often quoted 1% statistic. Except the 1% statistic is really based on aligned sequences of portions of the genome that we think are like the genome that's actually the code. All right, the stuff that really makes that really makes things. Right? Not the bunch of repetitive sequences and chunks of sequence that I don't need at all. Obviously, if I'm missing 300 million base pairs and I'm as functional as you are, those 300 million base pairs, they don't really make a difference, right? They're not the thing that makes us different from one another. And so you don't need to talk about them when you talk about the differences. What is the difference between organisms? We generally default to, or what has been defaulted to, is the aligned sequence between two organisms. Now, you can get to hey, there are some genes that one organism doesn't have or something. Well, you should count those as real differences, like the fact that there's a gene over there that this other organism doesn't have, right? And you can't align them because one exists and one doesn't exist. But that's a really small percentage of the differences between species, like between a chimpanzee and a human. It's true that large portions of the genome are only about 1.5 genetically different from human genome, right? So if you line up millions, hundreds of, if you line up 3 billion base pairs, because we have about 3 billion base pairs of alignable sequence between our genomes, which is 97 or 98% of the overall stuff. So we do share like that much of the genome. And then you ask how many differences are there in that sequence? It's about 1.5%. Okay. And between you and me, right, it might be 0.7%, right? 0.5%, 0.7%, maybe 0.8%, somewhere under 1% for all the sequence that you and I share between each other, especially all the sequence that's not the repeat sequence. The stuff that we've, if you sequence a genome and the part that doesn't get sequenced is all that repetitive stuff that just repeats the same thing over and over again. You might have more repeats than I do. And that's why the percentage would go higher if you started adding all that stuff up, and depending on how you count it. And just in case, 14.9% doesn't sound like very much to you. No, it sounds like an enormous amount, all right? But it's because of the way it's being counted. Consider that represents hundreds of millions of differences, and evolutionists only have 6 to 8 million years in their worldview to account for that vast number of differences. I'm sorry, did you just hear me say that there are people on Earth today that have several 100 million differences between them? Right? And they've only had 6,000 years. All right, since they had shared a common ancestor, and presumably the common ancestor would have had the same size genome. So that's, 6, 7, 8% difference in humans. Again, don't hear me wrong. Human beings are incredibly similar genetically. We're very homogeneous compared to a lot of species. A lot of species have a lot more variation. I'm pretty sure that, I know from sequencing other genes, chimpanzees have more variation between any two chimpanzees. Now, remember, we've only sequenced this one chimpanzee. You have to ask, what's the difference between different chimpanzees? And I don't know the answer because we don't have complete sequences that are as good as this sequence between two chimpanzees. But the difference between a chimpanzee and a bonobo, which are really similar species, most people consider them, barely different species, right? According to this analysis, I remember from that paper how many million base pairs is different, but it's much more than any two human beings are. Right. So it's probably more on the order of like, 8 or 9%, maybe 10% difference using the same valuation. And yet Ken Ham is very quick to say that bonobos and chimpanzees are the same kind. In fact, the ARC encounter says that gorillas, chimpanzees, and orangutans and bonobos are all the same kind. They come from a common ancestor. They come from just two individuals. And the amount of difference between a gorilla and a chimpanzee is greater than a human and a chimpanzee. All right, so it's more than 14%. It's like, it would be more like 20%. And yet Ken Ham is like, oh, they're the same kind. They came from a common ancestor. Wow, so there's an interesting evolutionary assumption that Ken Ham has, that they share a common ancestor, and yet they've accumulated 20% difference in their genome using the way they're doing the statistics here. I don't know. I mean, I'm sorry. I'm getting a little bit excited, but this is just how ridiculous sometimes the young earth creationist literature is, in that they do not present apples to apples comparisons. Right? This is all a shell game of using a particular way that sequence similarity is calculated and the 1% value that is being heard all the time. And then trying to like take out this other completely different way of doing comparisons and say that somehow, scientists have been lying before, or that this is a really significant value. The 14.9% represents something that's so vastly different that it should be shocking to everybody. All right, then we've got Georgia Purdom, who is an Ohio State PhD genetics, works for Answers in Genesis, although she hasn't done any genetics in a long, long time. What did she say? What most people don't realize is the oft-quoted 1% difference between human and chimp genomes only represents one type of difference, substitutions in aligned regions. Okay, that's what I was just explaining. Why only count this type of difference and not the others? Well, we do count the other difference. I mean, there are other people who have calculated differences. It's just that those numbers are never been considered very meaningful when you're like comparing other species. Like if I want to say like give out a number in terms of similarity between a fox and a coyote, or my house cat and Siberian tiger, right? The genomes can vary wildly in terms of size because of all this repetitive sequence and jumping genes and all these other things that make genomes increase and decrease in size quite rapidly. But those don't represent the real differences between those organisms. Genome size is not what it's about. It's about how you use the genome and what is the information content, I'll say, of the genome. And that would be the things that are actually being used to make those organisms. How similar are those things? Right? And that's the thing that most of us want to compare. I mean, there are deer species that you probably couldn't tell a difference between the two. You might not know they're different species, for which one has a genome that's twice the size of the other species. Twice the size. Right? So that, according to this kind of calculation, would be what, a 50% difference? 50% difference, which to the person who doesn't know what they're talking about, I mean, to the reader who doesn't know like how this number is calculated, they're thinking, oh, if I had a sequence of 100 base pairs, 50% of them would be different. How could you even be similar if you had 50% differences? But of course, that's not what they mean. They mean like one of them has just a whole bunch of copies of sequence that the other one doesn't have. It's repetitive. I've got 1 million copies of this and you have 100. and your 100 doesn't even do anything. Right? So that difference is not a meaningful difference. Why should we talk about that difference and try to calculate it? The calculation would give you a meaningless number like this 14.9% thing. It's a very meaningless number. especially if you're not going to do the pairwise comparisons and compare like human to human, human to Neanderthal, chimpanzee to bonobo, chimpanzee and show the matrix. Show me the matrix. I never see that matrix in Young Earth Creationist material. Yeah, I don't think it's really worth reading her comments. This is a massive blow to evolutionary worldview, as the supposed similarity between chimps and humans is touted one as the strongest proofs of evolution. Again, making it sound like to their audience, like this kind of difference is so great that you couldn't possibly have a common ancestor. But as I've already pointed out, they believe in thousands of species for which they're as different, if not more different from one another, that have common, that they believe have a common ancestor. So how is it a blow to evolution, the evolutionary worldview? If they're just going around saying, oh yeah, those things are all, you know, donkeys and horses are the same kind. They came from a common ancestor. And yet those two organisms, I'm sure, are more than 15% difference if you did this type of calculation, right? They have different chromosome numbers, right? The chromosomes are different lengths, right? So you're going to, obviously they have different total amounts of DNA. And so when you do the calculation, you're going to get a big number in terms of the difference if you're just doing it this way. Maybe that's why Nature didn't make a big deal about the researchers' stunning findings. Yeah, they published the paper. It's in Nature. Any paper in Nature is a pretty big deal. Was it like earth-shattering on the front of newspapers everywhere? No, because it was not stunning. It was not shocking. It was like, hey, we finally finished the whole sequence and we found exactly what we expected. We knew that there was big differences in the amount of total DNA, and we knew that there was going to be a repetitive sequence, and we knew that it was really hard to sequence in the past. And with new technology, we got the sequence finally. And now we can talk about how, like, more data about how these are different from one another. Right. Okay, so we've reached, let's see if there's anything at the end here. Once again, observational science confirms the Bible. Chimps and humans aren't related. The ape kind was created by God as a separate, distinct kind, as were humans. Did you see that? The ape kind. This is just ridiculous. What else can I say about this? Make sure you follow me here, especially if you're a Young Earth Creationist watching this. They just said the ape kind, and I just told you a minute ago, there's a display at the Creation Museum that shows two apes getting off the ark. And those two then speciate into chimpanzees, orangutans, bonobos, and gorillas, plus a bunch of extinct versions of apes, including Ardipithecus and some of those other supposed hominid, human-related things that were not standing upright, that they don't consider humans. They're actually considered to be the ape kind, which means they all share a common ancestor. And I guarantee you that those genomes between all those different species are more different than between a human and a chimpanzee. And yet Ken Ham's saying they're the same thing. They're all related by common ancestry. Just 4,350 years ago when they stepped off the ark, the two apes that stepped off the ark. And their genomes changed so much that they became more different than a chimpanzee and a human genome. Just in terms of the raw sequence and the code and everything that was there. Most of their audience is not gonna see that. They're not gonna figure that out. They're not gonna realize that they're the ones that are having the wool pulled over their eyes. Ken Ham is deceiving his audience. Now, that's a pretty strong word. I'd say he would be deceiving it if he understood that himself, which he should, because somebody should have told him about the numerous videos I've made that have made this point over and over and over again, and many others have made this point. In other words, he's without excuse. He can't say, well, I didn't know that. He should know. I mean, he wrote this blog post, right? I mean, I don't think he really did, but let's see if it says it. Yes, this item was written with the assistance of AIG's research team. But he takes ownership for this. So he's acting like he's read the original paper. And again, I don't think they read the original paper. I think they read the Evolution News article and they're trusting that. So they're trusting a bad source. And then they're making it worse. We've actually made the main points, but now we just want to go and I want to reiterate them by looking at the actual papers. Look, before I go to the Nature paper, let's just zip by the National Human Genome Research Institute, where I was reading up on human genomic variation. The vast majority of DNA letters in people's genomes is identical, but a small fraction of those letters vary. Now, vary would be, look over here, like you have a C and a T, or you might have one parent gave me a C, the other parent gave me a T. So I have a C and a T, right, in my 2 copies of my genome. You might have a CC or you might have a TT, all right? That's your basic genetics, like where your variation comes from in your genes, all right? And so, we're all going to have small differences like that. And when you add that all up, again, that's way less than 1% sequence difference. But there's a lot of other types of differences between us. But this is the kind we're most interested in, or just like an insertion of a base or a deletion of a base, or some other shifts around. But it's basically small stuff. I can show you. All right. We each got 1 copy of each of our chromosomes from each parent. So that's why we have 2 copies of all of our chromosomes. And your 2 copies might not be the same length. Right? Because your mom's chromosome might be a little longer or a little shorter than your father's, which means that they have a different amount of sequence, which means that if you add the amount of sequence difference they have and you count every one of those as a difference, you're definitely going to have more than 1% difference between you. But again, you're probably going to have all the same genes. Like we all have the same genes. The differences in terms of size is areas outside of genes. It's the spacers between them that increase in length and decrease in length. And if you add those differences, you're going to get a lot more dissimilarity between you and me and everybody else. But if you're just looking at the A, T, C's, and G's that are variants between us, then very small amount, right? 99.9% identical to each other. I mean, this is the common parlance. This is the common way that we talk about genome variation. All right. A single nucleotide difference exists between 2 people's genomes once every 1,300 nucleotides or so. Now, this side is right. And this is where, everybody should do a little extra reading, right? To find out like some of the caveats. However, this is an oversimplification because it accounts for single nucleotide differences. In reality, 2 people's genomes are on average 99.6% identical, or about 0.4% difference. That is, remember, you have 2 copies of your genome. And they're not identical to each other. So even in you, there are differences between the 2 copies of the genomes you have because you got them from 2 different parents, right? And they're more like 0.4% difference, right? So that's 3 times that. So once every 800, once every 500 or so base pairs. All right. So the differences among human genomes we call genomic variants, right? And it's those variants that make us unique. I mean, after all, if we had exactly the same genome, all right, then we'd be identical twins, right? We'd be morphologically, physically, we'd look almost identical to one another, right? The environment has some effect too, right? So it would be absolutely identical. So then they're just demonstrating the types of variants, right? Single nucleotide variants, individual base pairs that are different. Again, those are the ones we really care about the most. We do care about small insertions, small deletions, and then we have things called tandem repeats, CAG, 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 CAG. And you might have, and this is what I'm saying, maternally, I might have 3 copies of that repeat, and my dad gave me 4 copies. But my mom gave me 4 copies of this other repeat, or this other person has 4 copies, and somebody else has 2. By the way, this is microsatellite analysis. We use it for fingerprinting because at many, many different sites across all your chromosomes, you have different numbers of tandem repeats at locations that we know of. And so we can look at, like you look at 20 different locations and you have 2 copies, one for your mom, one for your dad. You might have different variants at each one of those sites. You get a profile of somebody. It's like, hey, I got 2 here, 4 there, 6 there, 10 there, 15 there, 25 there, and so forth. The chances that another person not related to you has the exact same combination is very, very small. So therefore, if you have the same DNA fingerprint, the same tandem repeat profile as somebody else, you probably had to get them from the same parents. right? The chance, all right, that happened by chance is so small that in a court, right, you're going to probably convince a jury that those two people have to be related, right? They have to be identical twins. They have to be close relatives in order to have such similarities in those tandem repeats. But the point is that if you add it up, you could have thousands and thousands and thousands of 3, 4, 5 base pair repeat differences. And if you have 1,000 5 base pair repeat places, that's 5,000 base pairs that you and I differ at. And that's just size, right? You have 5,000 base pairs. Maybe I don't have at all. So you start adding those things on, and tandem repeats are just small repeats. There are many other much larger repeats. We have whole duplications of whole regions. I might have copied 75,000 base pairs and have 2 copies of that 75,000 base pairs. So I potentially even could have 2 copies. There are people that have 2 copies of some genes that somebody else only has 1 copy of. All right. Just like chimpanzees have copies of genes, like more copies of some genes than we do. And we have more copies than they do for some genes, right? There's been duplications. If you add all this sequence up and say, well, all of this doesn't exist in the other one. And so those were, if this is 100,000 base pairs, that's 100,000 differences. Now, other geneticists might say, and I would tend to fall in this camp, the repeat is a single change. That's a single difference between 2 organisms. It's exactly the same sequence or nearly identical sequence. And the difference is a one-time event where it got duplicated. Somebody had it duplicated, right? Your mom's might have 1 copy of that 100,000 base pairs, and your dad might have given you 2 copies of that 100,000 base pairs. That doesn't mean for that region, if you just had that 100,000 base pairs, you're like, I'm 50% different than you because I have 100,000 more base pairs. So I have to divide, my 200,000 by your 100,000. But that's what they're doing here. That's what Ken Ham is doing. I don't think he knows that's what he's doing, but he's counting every single difference in this duplication. And sometimes inversions mess that up too. All right, there's other things that happen in the genomes that end up creating differences between different organisms. I mean, if you have inversions, insertions, translocations, all of those could be counted up as differences, even if they don't functionally make somebody operate differently. Yeah, there was a data here. Okay, yeah. So what does a typical human genome sequence look like with respect to the variants that it contains? So we're talking about within human beings, just comparing across different individuals. This is important to get a feel for, so you're not hoodwinked by people trying to show you how different species are, as if that's some, like, don't let it be more radical than it is. Different species are different. They are expected to be more different than individuals within a species. But don't get caught up in the craziness of 15% difference. I mean, you and I may have 5 million, all right, nucleotide differences, single nucleotide variations. And then, as I said here, there could be 600,000 insertion and deletion variants, identified in human genomes, right? That would be 2 million different nucleotides involved, right? Potentially a 2 million base pair difference between us, depending on which variants you have and not have. I mean, you might have fewer differences of that 2 million, but somebody has 2 million differences between 2 individuals. All right, so you put that's 7 million. And those are not the ones we're usually using when we talk about sequence difference. And then there are structural variants up to 20 million nucleotides. And then there's the case of extreme changes of duplications, all right? Right, adding big chunks of sequence, which I was saying potentially 100 million, 200 million, even 300 million base pairs. So when you add all that up, right, then you're getting into the range of, hey, you and I very well might be, by that kind of form of calculation, or you're counting every possible difference, you might be 3%, 4%, 5% different than me, or in the extreme, maybe 8, 9% different. Ken Ham would say that should be then shocking, right? I mean, 9% difference. when scientists have been talking about how you're 99.9% similar. Right? But here, right here on these sites, from National Institutes of Health, US government sponsored stuff here. This has been saying this for a long time. This isn't brand new. They didn't change it because of the paper. This already existed. We already knew 20 million base pair differences between individuals, 100 million base pair differences between individuals. Okay. This is not shocking. I'm just going to keep saying that. It's not shocking. Let's take a quick look at this paper, because I've thrown out the 300 million number, so I want to show that to you. I didn't just make that up. Telomere to telomere, assemble a diploid reference genome for Han Chinese. All right, telomere to telomere would be end-to-end chromosome sequence. This is the new technology that allows us to read long sequences and blow through these like really long repeat sequences. You have to have a long read in order to figure out whether you have 50, 100, 1,000, 10,000 repeats of the exact same sequence over and over and over again. That's what was so confusing in the past about the human genome. There were sections where we knew it's like, you got a lot of repeats there. I just don't know how many. And again, we didn't really care that much because it's like, okay, you got 10,000, I got 5,000, someone else has 25,000 repeats. Does that make you different than me? No. Very unlikely that it's going to make any significant difference. I'm not saying it never makes a difference. Sometimes there can be structural things about the chromosome that change some regulatory pattern. All right, so let me just give a quick read of this abstract. Since its initial release in 2001, the human genome has undergone continuous improvements in quality. Absolutely true. Constantly trying to get a better sequence. The recently released telomere to telomere version, T2T CHM13, reaches its highest level of continuity and accuracy after 20 years of effort by working on a simplified, nearly homogeneous individual of a hydatiform mole cell line. Right, that's the new human genome that was sequenced that's considered the most complete sequence. But remember, that's one individual. One individual does not represent the human race. It represents that one individual. Here, to provide an authentic, complete diploid human genome reference for the Han Chinese, the largest population in the world, we assembled the genome of a male Han Chinese individual we're going to call it telomere to telomere YAO, which includes all of the 22, that would be your 22 autosomes, the X chromosome, and the M, which is the mitochondrial genome. And then they also did 22 chromosomes, autosomes from another individual, and the Y chromosome, right, from the male. The quality of T2T-YOA is much better than those of all currently available diploid assemblies, and its haploid version reaches top quality of fewer than 1 error per 29 million base pairs. That's how confident they are in the sequence that they have. Even higher than the other one that was reported, right? Okay, now, each haplotype of T2T-YO possesses 330 million base pairs of exclusive sequences. Each haplotype. 3,100 unique genes and tens of thousands of nucleotide and structural variants as compared to CHM13, which is the other sequence, is the original best sequence for the human genome, highlighting the necessity of population stratified reference genomes. Did you catch that? 330 million base pairs of exclusive sequences, including 3,100 unique genes. Most of these genes aren't like big genes that make proteins. They're probably talking about little microRNAs and a bunch of other regulatory stuff, right? Nonetheless, it's saying that within 2 human genomes that have been sequenced, right, from 2 different populations, we have 300 million base pairs of unique sequence difference between them. And we even have genes that are different between them. Yeah, have you got a gene? I don't have that gene. Right? That's the variation that exists in human beings, which is, I'll keep saying it because I just want to make sure you understand. As a species, human beings are relatively homogeneous. We don't have a tremendous amount of genetic variation, which tells you that other species that we would consider more genetically variant would have even greater amounts of variation, right? even more differences in numbers of genes and total amounts of sequence. Right? So imagine that you sequence representatives of some of the most ancient lineages of modern humans from Africa. Right, and we get really good sequence from those. We have some sequences, but we don't have T to T. All right, telomere to telomere, and that's what you need to do to get all that spacer sequence, all that repetitive sequence. All right, so what we do have suggests that yes, they're a little bit more different, but it's still within the 1%. 99% similar. But that's for all the sequence that we have in common, all the common genes that we have, not all that other stuff. Right, that often makes individuals different from each other in terms of just the amount of sequence they have. All right, so that's an enormous amount of variation. Again, Ken Ham should be like absolutely stupefied by that amount of variation if he thinks that this chimp human thing is like a stunning revelation. He should be absolutely stunned by this particular paper. This is December 2023. All right. Why wasn't this headlines everywhere in the world? Because it's like, okay, yeah, there's that much difference. Am I that different than a Han Chinese? No. In terms of real significant differences, no, I'm not 9% different. It's like that percent doesn't mean much. Okay, we're almost done here. So this is April 2025. Again, lots and lots of authors sequenced all these different genomes. Today we're sequencing, I don't know how many hundreds of genomes, of whole genomes of organisms are sequenced every single day now. But this is a much higher quality level of sequencing, but for a complicated species. By the way, there are some organisms like lampreys and coelacanths and things like that have genomes that are like way bigger than the human genome and have like so much repetitive sequence in them that they're very, very difficult to sequence. Nonetheless, the human genome's pretty darn big, but it's very similar in size to all the other primates. All the other great apes. The other primates have more differences than this. And then if we just scoot down here, I just want to give you one visual example of what's going on here. So let's look at here. This is chromosome number 16. And across the bottom here is MBs, which is million base pairs. So you have 125 million base pairs. That would be if you strung out the entire chromosome 16 DNA content. Remember, chromosomes are made of proteins and DNA. So just the DNA part we're talking about. And then you would align that up. You see like HSA is human and PTR is pan troglodytes, right? That's chimpanzee. And so you line those up and all this gray stuff here says we're nearly identical. I mean, there could be individual nucleotide variants in there, A, T, C, G, but nothing big enough that you would see it when you're looking at this level of 150 million base pairs. I was down chromosome number 16. So you're scanning across here, you see a lot of gray, and there's little yellow spots here, and those are inversions, meaning that the sequence in the human being, if I took that sequence and I looked for it in the chimpanzee, I'd find out it's in the opposite direction. It's inverted, turned around, facing the other direction. It's the same essential sequence. The sequence is probably 98% similar, 98.5% similar, that 1.5% difference, because it lines up with the human sequence. It's just in the opposite direction. Now, if Evolution News wants to count that as, that's 5 million base pairs that are all in a different direction, so that makes them different, well, then their percentage is going to keep going up. They're going to say, oh, that's a huge difference. The thing is, if there are 5 million base pairs running in a different direction, it doesn't change how the genes work. They can still be read in the other direction. So you flip those around, it may not make any difference between a human and chimpanzee that they're flipped around. So you have a bunch of little tiny inversions, little tiny segments that are like flipped around between the two genomes. But you'll see that between human and pan, and this is bonobo down here. And so you see bonobo and the bonobo and the chimpanzee, which are, for many people, they feel like they're the same thing. A lot of them call it the pygmy chimpanzee, small version of a chimpanzee, but it's considered a different species. You'll see that it's very similar, right? It lines up really well with each other. right? But look at this. The Pongo is much bigger, right? It has a bigger chromosome by a good bit, right? We're talking 10 million base pairs longer. So that's 10 million base pairs difference right there, just in one chromosome between two chimpanzees that most people feel like are really close sibling species. And Ken Ham definitely thinks those are the same kind of organism and came from a common ancestor. But now go back up to humans. And most of it's because there is a large gap here. I hope I read that right. I hope I'm reading this graph right. I apologize if I'm not reading it right in terms of this, the amount of sequence difference between the chimpanzee and the pongo. But I do know that I have it. I do know I understand the part between humans and chimpanzees. So let's go there. Let's go there then. Right here, there's this pink bar right here. Those are satellite sequences. Satellite sequences are repeat sequences from like jumping genes, right? Or they could be from viral insertions or things like that, right? Horizontal gene transfer from another organism, right? So, and this would be probably all of us have something like this, right, in us. That is a huge chunk, right? So that there, right there, you can see right away that there is some 10 million base pairs that the chimpanzee does not have. So you and I have 10 million extra base pairs. There are no genes in here, right? There's no genes. It's all satellite sequence, repetitive sequence, no genes, not really being expressed, turned into things, doing anything. It's just stuff stuck into your genome. But Ken Ham, Evolution News, they want to count that as 10 million differences. Those 10 million differences are not equivalent to the individual variants that are different between us in the same area of sequence, because those individual variants are in genes that make you have a different amino acid than a chimpanzee, which makes the gene function differently. That's what we care about. That's what we're talking about. Where all this gray stuff is, where we're aligned, and actually including the inverted repeats, 98.5% similar, right? And then you can increase that distance or dissimilarity by looking at these chunks. Centromeres are a gob of repetitive sequence, and the centromeres are slightly different. You can see like the centromere in this Pongo orangutan is far larger than in human beings. All right, so there's a big difference there. And look at the differences between chimpanzees, gorillas, and down here, the orangutans. These two species of orangutans are very similar to each other, right? They're much more similar, all right, than other species are. But look at the differences between these different primates. I mean, especially here. I mean, the gorilla compared to the orangutan. And here's another big difference. Here's a subterminal heterochromatin. All right, so this is a bunch of additional sequence that the pongo has versus the chimpanzee or the pygmy chimpanzee, right? Big difference in sequence there as well between two really closely related species. I know I'm beating a dead chimpanzee on this at this point. This graphic down here kind of tries to give you an idea of what the genomes look like in terms of the type of sequence. You can break down a genome into, you got your telomeres, which are repeat sequences at the end of your chromosomes. You got your centromeres, which is where your chromosomes connect to each other during like meiosis. That's a lot of repetitive sequence too. Many, many tens of millions of base pairs of sequence. But some people have, some individuals, some species have different amounts. So let's just take a look at this part of this figure right here. We have mobile element insertions. And they've measured that for how many insertions there have been over millions. Insertions would be like added sequence to the genome from external sources, of jumping genes, basically. Let's just take a look at this portion of the figure, mobile element insertions. So these would be sequences you have in you that have the capacity to replicate themselves, jumping genes, or you call them selfish elements. They can copy themselves and insert themselves in another part of the genome. So sometimes it's tandem, meaning next to each other, and sometimes just like, hey, I just pick myself up, copy myself, and put myself somewhere else. And it's showing different primates and different elements. So line 1 elements, SVAs, ALUs, which is really common short sequence that gets repeated. And you see humans have, a large number of these ALUs, suggesting every million years they pick up 1,000 more copies of them. Very similar rate to the chimpanzee. But other primates have different amounts of other types of inserted elements. So it's all these types of things which are really non-functional DNA. I mean, they're copying themselves. Sometimes you have more copies. You have thousands of copies. Again, you have more copies or less copies of ALU elements than I do. So because of our history. I mean, if you're very closely related to me, then we probably have about the same number of ALU elements. But if you're distantly related to me, then you've accumulated ALU units because in your ancestry, you might have had some copy themselves and I didn't have them copy. Or maybe I copied more than you did, right? And so we're going to get a different number of those, which is going to do what? Means we have a different size genome as a result. So all of these things is just to say, I don't want to get into all the details on those, but I don't think I need to do more than just point out that we have a lot of differences. But the question really becomes, what are the important differences and how do you calculate those important differences? How do you calculate the difference in sites that matter? And use that as an apples to apples comparison of different species and say like, oh yeah, you're about that % difference. This is why we like to use the mitochondrial genome sometimes, because it's a little more clock-like and it's like a smaller genome and it's easy to see the differences between organisms. All right, I think I'm going to quit there. Yeah, that's my quick assessment of chimp DNA not being 99% identical to ours, but hey, big whoopee. This is not really a story. This is not something that Ken Ham, I mean, this is a great story for Ken Ham. They've been making a prediction. Oh, it reminds me a lot of Nathaniel Jeanson, the gold standard predictor, right? He likes to make a lot of predictions and then test them. All right. Hey, if you're going to make a prediction that the human and chimpanzee genome is not 99% similar, you would have already known. I already knew that it wasn't. All right. That's not a difficult prediction to have come true if you're going to calculate it in a different way. Right. It's just not. Okay. I've been going on far too long on this. Thanks for hanging with me. Yeah. We'll talk to you later. Bye-bye.